Excretory products and their elimination. Practice questions for NEET part 2. Let's go with the question here. Select the incorrect statement from the following. The DCT of many nephron opens into a straight tube called collecting duct. In cortical nephron, the loop of Henle is too short and extended only very little in medulla. In juxtamedullary nephrons, the loop of Henle is very long and runs deeply into the medulla. Vasa recta is not a part of peritubular network. So DCTs of many nephron open into straight tube called collecting duct. Okay. In cortical nephron, majority of nephrons are cortical nephron. They are saying yes, 80 to 85% are cortical nephron. The loop of Henle is too short and extended only very little in medulla. Too short or maybe absent even, we can say. In juxtamedullary nephron, minority, less in number. Okay. The loop of Henle is very long and runs deeply into the medulla. Yes, true. Vasa recta is not a part of peritubular network. No. Vasa recta, peritubular network are the extensions of the efferent arteriole, blood vascular system. They belongs to peritubular network in the cortex, associated with PCT and DCT, a blood capillary it is. Vasa recta is also a blood capillary associated in the medulla. The two kidneys of human being lie at the level of ovaries at the same level, left kidney at a higher level than the right one, right kidney at higher level than the left one. The two kidneys of human being, left kidney little higher level than the right one, we can say. Which term is used for both for a part of kidney and part of skeleton in the mammals? Centrum, pelvis, cortex and medulla. Part of skeleton and part of kidney. Cortex and medulla you might find somewhere else like ovary in the human reproduction, female reproductive system. Pelvis, centrum. Centrum is a part of vertebral column. Pelvis. Pelvic bone, we say. Mammalian kidney serve to excrete excess water, urea and amino acid, excess salts, urea and excess water, excess salts, excess water, excess amino acid, excess salts, urea and water. Mammalian kidney serve to excrete excess salts, urea and if it any excess water. The part through which arteries and veins enter or leave the kidney is called the major calyces, minor calyces, hilus, hilum we can say, renal port. So hilum. Cells named podocytes occur in the wall of neck region of nephron, glomerular capillaries, outer wall of Bowman's capsule, inner wall of Bowman's capsule. Podocytes are the part of Bowman's capsule. Okay. So here asking about outer wall or inner wall. So if this is the Bowman's capsule, then what we say, this is outer wall. Okay, and this is going to be the inner wall. So filtration is going to happen with inner wall. So that's why our so-called podocyte, that means what is podocyte here? The finger-like projections we can say. The finger-like projections will find at neck region of nephron, glomerular capillary, outer wall of Bowman's capsule, inner wall of Bowman's capsule. So what we have to say? The inner wall of Bowman's capsule. It is there at the inner wall of Bowman's capsule. Okay, then a Malpighian corpuscle is another name for nephron, an excretory structure of insect, and combined name for glomerulus and Bowman's capsule. None of the above. So combined name for glomerulus and Bowman's capsule. This is the Bowman's capsule, and into the Bowman's capsule our afferent arteriole enter inside here, afferent arteriole, and form the so-called glomerulus, and will go in the form of efferent arteriole. So the total setup we are going to say it as combined name for glomerulus and Bowman's capsule. We say it as a Malpighian corpuscle. Malpighian corpuscle. Okay. So then next. Blood vessel leading to glomerulus is called. Blood vessel leading to glomerulus is called. Let's talk about this is the Bowman's capsule and who is coming into the here. Afferent arteriole is coming and it forms the glomerulus, and this is leaving the Bowman's capsule. We have to say efferent arteriole, and this is afferent arteriole, we can say, leading into the glomerulus. So what we have to say? Afferent arteriole. This is the afferent arteriole. Okay. So then next, blood vessel draining the glomerulus in a mammalian nephron is called. Blood vessel draining the glomerulus in a mammalian nephron. Afferent arteriole and is narrower than the vessel entering it. Efferent venule and is narrower than the vessel entering it. Efferent arteriole is narrower than the vessel entering it. Renal artery and is wider than the vessel entering it. Asking about blood vessel draining the glomerulus. Talking about this one. So again, let's draw the Bowman's capsule. This is Bowman's capsule. So a blood vessel entering into the Bowman's capsule in the form of glomerulus is afferent arteriole. Then leaving the glomerulus means we have to say efferent. So what is our answer then? Efferent arteriole is narrower than the vessel entering it. Here it is. And draining the glomerulus in a mammalian blood vessel, draining the glomerulus in a mammalian nephron. So we need to answer. If venule, there is no matter of venule here. Wherever venule is there, you should remove. Renal artery enters into the kidney. So let us eliminate this also. So these two are the answers. We have to find out afferent arteriole and is narrower than the vessel entering it. Vessel entering itself is the afferent arteriole. Here, who is entering itself is the afferent. So then that's why we say this is also wrong. So finally what is our answer? Efferent arteriole, which is narrower than the vessel entering it. It means here you have to understand the afferent arteriole is wider and who is leaving, that is efferent arteriole, leaving the Bowman's capsule, that is going to be, this is narrower and this is wider. Afferent is wider and efferent is narrower. Okay, moving on. In a mammalian kidney, the Bowman's capsule or Malpighian corpuscle occur in mammalian kidney, Bowman's capsule or Malpighian corpuscle occur in cortex, medulla and pelvis, all of these. So if it is the cortex and this is medulla represented in the form of renal pyramids. So let us see our Bowman's capsule, that is in cortex, and PCT in the cortex. Henle's loop, the longer Henle's loop in juxtamedullary nephron, present in the medulla, whereas DCT, distal convoluted tubule, in cortex. Collecting tubule contain both cortex and medulla. So what's the question here? Bowman's capsule or Malpighian corpuscle. So where it is? This is in the cortex. Where it is? This is in cortex. So that's it. Our answer is cortex. Then next. In a glomerulus, afferent arteriole is thicker than efferent arteriole. Afferent capillaries are thicker than efferent capillaries. Afferent capillaries are thinner than efferent capillaries. Afferent arteriole is thinner than efferent arteriole. Okay, so what's it? Afferent arteriole is thicker than efferent arteriole. Afferent arteriole is thicker than efferent arteriole. Okay, next. Which one of these is not a part of uriniferous tubule? Loop of Henle, collecting duct, Bowman's capsule, distal convoluted tubule. Which one of these is not a part of uriniferous tubule? Who is not the part of uriniferous tubule here? The collecting duct is not the part of uriniferous tubule. Bowman's capsule is lined by squamous epithelium, ciliated epithelium, cuboidal and columnar. The Bowman's capsule, outer and inner walls are composed with the simple squamous epithelium here. Simple squamous. This is the one. You can see that if you show here, this is a simple squamous epithelium. Bowman's capsule composed of with little podocytes. That means here like this. If you see outside, it is like this. This area I am showing here. So the cells look like food, and so we call podocytes. Bowman's capsule is lined by. What's our answer then? Squamous epithelium. Simple squamous epithelium arranged as podocyte. Brush border is the characteristic of. The Bowman's capsule, as we said now, made of simple squamous. Here it is the simple cuboidal epithelium. We can say here, this is the simple cuboidal epithelium. The surface of simple cuboidal epithelial cells of PCT is having that folds like this. We say it as brush border. So what is our answer? Proximal convoluted tubule. Filtration of blood occurs in. Let me show you. This is a Bowman's capsule. Our afferent arteriole came here in the form of glomerulus and going out in the form of efferent arteriole. So where does the filtration of blood occur? Here it is. This is the filtration. Blood capillary came to give excretory wastage. Our Bowman's capsule is ready to take. So what is our answer? Loop of Henle, Bowman's capsule and lungs. So our answer is Bowman's capsule. The glomerular filtrate, that is the liquid collected in the cavity of Bowman's capsule, is the glomerular filtrate, that the liquid collected in the cavity of Bowman's capsule, whatever collected here in that place, that all the components of plasma, blood plasma, except proteins which comes here, and also RBC. We can't expect RBC and WBC also. So that's why our answer is blood minus proteins and corpuscles. Normally, that is absent in glomerular filtrate. Normally means here after the filtration, the excretory wastes from the blood are coming. Yes, sodium chloride will come, glucose will come, creatinine might come, then a protein, albumin is the protein, so I can't expect albumin in the filtration substances. The glomerular filtration rate would be decreased by constriction of the efferent arteriole, an increase in afferent arterial pressure, compression of the renal corpuscle, an increase in the renal blood flow. So compression of renal capsule, the glomerular filtration rate would be decreased by compression of the renal capsule. That means renal corpuscle, we have to say here, the capsule is not much suited word here. So that means vasoconstriction they are saying about, vasoconstriction. A minute vessel run parallel to the Henle's loop forming U-shaped vasa recta is a part of peritubular network, afferent arteriole and Bowman's capsule. So what is our answer then? Is a part of peritubular network. So let me show you here. This is the Bowman's capsule, PCT, Henle's loop and DCT, and this is the collecting duct. So just I am showing all of these in black color, and let me show you the blood vascular system here. Afferent arteriole belongs to the blood vascular system. Glomerulus, and above on the PCT and DCT it form the peritubular net. Above on the Henle's loop it form the vasa recta. So all this is a part itself, a part of blood vascular system, we can say. So peritubular net, we can say. Okay, then urine formation mainly involves the process of ultrafiltration, selective reabsorption, secretion. These three are the methods of urine formation, we can say. Next, how much amount of blood is filtered out by kidney per minute? 500 ml, 1100 to 1200, that 1500 ml and 125 ml. So the total story we have to find out like this. This is the heart. From the left ventricle of the heart, we know that nearly 5000 or 5 liter water will come here. 5 liter water. So let me write here like this. This is the 5 liter water, and among the 5 liter water, talking about blood here. So 5 liter blood will come out and it will go into renal artery. So how much it is going into renal artery into the kidney? 1100 to 1200 ml. Okay, so total cardiac output is nearly 5 liter. In that, entering into the kidney is 1100 to 1200, we have to say. How many layers do filtration membrane consist of? Filtration membrane, this is Bowman's capsule and afferent arteriole, glomerulus, efferent. So this blood capillary endothelium of the blood capillary made up of simple squamous, whereas our Bowman's capsule made up of simple squamous. In between them we say basement membrane. So that how many layers can be expected then? Layers to filtration membrane consists of 3. Okay. Filtration membrane consists of, as we said now, as we are shown now, filtration membrane consists of endothelium of blood capillary, a basement membrane, and the simple squamous epithelium of Bowman's capsule. So what is our answer here? Endothelium of glomerular blood vessel, okay, and epithelium of Bowman's capsule, okay, basement membrane between 2 layers, that also okay here. Then all the above is the answer. Next, which of the following is incorrect about ultrafiltration? Podocytes are arranged in intricate manner so as to leave minute spaces called filtration slits and slit pore. Filtration occur finely through the pores. Okay. Filtration is so fine that almost all the constituent of blood except protein pass into the lumen of Bowman's capsule. Filtration fluid is isotonic to blood plasma. JGA controls the filtration rate of ultrafiltration. Okay, so what is our answer then? Filtration is so fine that almost all the constituents of blood except protein, okay, except protein and blood corpuscles, they also don't come here. RBC and WBC, they also don't come. The values of GFR in a healthy individual is, so what is GFR? This is the Bowman's capsule, as I already said, from the heart which is coming, that is called cardiac output. That from there renal artery 1100 to 1200 ml per minute is coming. Then afferent arteriole, we are talking here now. The values of GFR, glomerular filtration rate. We have a value here that is 5 liter, and we have a value here entering into the kidneys, as we said now, 1100 to 1200. Here it is 125 ml per minute. 125 ml per minute. Amount of the filtrate formed by the kidneys are, that 125 ml per minute, 7.5 liter per hour, 180 liter per day, that okay, per minute, per hour, per day, we get calculated. Select the total number of correct matching. JGA, juxtaglomerular apparatus, okay. Then glomerular filtration rate, GFR. PCT, proximal convoluted tubule, we can say. DCT, distal convoluted tubule. Conducting duct, collecting duct is better to say. Antidiuretic hormone. Okay, so then what's our answer then? How many are correct here? 1, 2, 3, 4. JGA is formed by part of DCT, part of afferent arteriole, both A and B, none of this. Let me show you here. This is the Bowman's capsule and afferent arteriole, this is afferent arteriole, glomerulus, and this is the efferent arteriole. Then distal convoluted tubule connected to afferent arteriole is going to form juxtaglomerular apparatus. So who are the participants here? The participant, one is DCT, distal convoluted tubule, and the other is afferent arteriole. So what is our answer then? Both A and B, part of DCT and part of afferent arteriole. Following are the points of mechanism of JGA, arrange them accordingly. Activation of JG cell, activated JG cells to release renin, fall in GFR, increase in glomerular blood flow, GFR back to normal. So I think we should start with fall in GFR, isn't it? There is a fall in GFR. Okay, when glomerular filtration rate falls down, activation of JG cells, where we are, we get started from C. So C, after that A, I think we are here. C, A, B, activated JG cells release renin, okay. Then a ROS mechanism will go on, that conversion of the angiotensinogen into angiotensin 1, angiotensin 1 into angiotensin 2 by angiotensin converting enzyme, and so on. It will go. Finally, what happens? So increases in glomerular blood flow that make GFR normal. So C, A, B, D, E. Our option is B. Okay, we continue with part 3 here, that it will go on like that, part 3, part 4, part 5, the practice of questions, and in each and every chapter that maximum practice required here for any student. So go on through this practice of NEET biology, excretory products and elimination. Part 3 will come soon. Thank you.